You're fine. Like a little like Diet blue.
I lifted last night, so I kind of
I liked it. No, I guess not. Yeah, it's like, you can.
All right, good morning, everybody. How was everyone's weekend? Yeah. Cool. All right, so today we will start off by going over the worksheet to review some answers. Did everybody get a chance to do the worksheet, like answer the questions to the best of your ability? For those of you who weren't here, you guys saw my message about that. All right, cool. All right, so first I will take attendance so that I can give you guys points for completing the worksheet. There's not going to be like a right or wrong answer. It's more of just participation and you trying and answering the questions and being involved in the discussion today to review them, because the goal was for you guys to understand the content. So, all right, Tegan, Lucia, Nicolo, Autumn, Ryan, Quinn, Celeste, Zoe, here, Annika, Jesse, Sarah, Jada, Clayton, Paige, Erica, Victoria, Brooke, Abby, and Hannah, Gabby. Juliana. Nick. Henry.
Here.
Tyler. Awesome. everybody is here. All right, so we could start going over the questions. I also have the other 3 lectures pulled up, so if you guys wanted to look at a visual or, to go through the concepts, I can pull those lectures up. So for number 1, why do you think so many environmental microbes cannot be cultured in laboratory broth or agar media? So I briefly talked about this, yeah.
Well, you said that like some of the environmental conditions can't be replicated in those mediums.
Yep, that's exactly right, exactly right. We don't have the specific environmental conditions and those things to actually grow those microbes, but how do we know that they exist? This is a second parter to this question. So we know that they exist from sequencing efforts. So there's been a lot of sequencing that's been done that tells us that there is DNA from these microbes, so we know that they exist. But we aren't able to grow the organisms in a culture or lab setting because we just don't quite know yet the actual conditions that are required. Remember, microbes are supposed to be really adaptable and can exist in very different conditions, and so it's hard for us to replicate them. Doesn't mean that we won't at some point. So that's also an important thing to remember there is we might not know at the moment, but we could eventually culture some of those organisms, but that's exactly it. All right, everyone good with number 1? All right, number 2, what are the differences between prokaryotic and eukaryotic microbes? Just a general question. Yeah.
Prokaryotes use smaller cellular and they do not have nucleus disorder.
Sorry, can you repeat that last part?
They don't have a nucleus to store the DNA.
So they have, yeah, exactly. So they are exactly no nucleus. They are typically smaller than eukaryotes, and some of them. One of the things is prokaryotes have some membrane-bound organelles, whereas eukaryotes have extensive membrane-bound organelles. That's one of those things that wasn't written in textbooks previously because we always thought that prokaryotes don't have any organelles, but actually that's wrong now. We know that they do. They just don't have as many. It's not as extensive as eukaryotes. So that's another important detail there. What about the nucleus for the eukaryotic organisms? How is that partitioned, I guess?
They're more like fixed in the.
Yeah, it's enclosed in a membrane bound. The DNA is enclosed in a nucleus. Yeah. Okay. What are some phenotypic features that distinguish bacteria from archaea? So we talked about how archaea are understudied. We thought for a long time, up until I think the 70s, that they were just bacteria or weird bacteria, but now we know that they're archaea, and so what distinguishes them from being bacteria or prokaryotes? Yeah. Either one of you guys.
The cell walls, they have different amino acids and sugars. Cell membrane is made up of different lipid structures and chemical links. And then archaea are more similar to eukaryotes.
Yeah, exactly. And the gene expression machinery is more similar to eukaryotes as well. Nice. Any questions on those so far? Okay. What are the 2 requirements that need to be met in order for algae to grow?
Water and sunlight.
Yep, exactly. Wherever there's like moisture and sunlight, algae will grow. Remember that polar bear analogy that I gave with the polar bears having green in their follicles because they were surrounded by moisture and sunlight, so algae was growing in the hair follicles of those polar bears. What makes fungi stronger than steel? Yeah.
The cell walls contain chitin.
Yep, exactly. Chitin. All right. So what are the benefits to the plant and to the fungus in a plant mycorrhizal symbiosis? Yeah, Henry.
It extends the volume of soil the plant is able to reach.
Yep, so that's something that the fungi will provide for the plant. That's right, and so it helps to extend the volume of the soil accessible for the plant. What structure allows the fungi to do this? Yeah.
Mycorrhiza.
So the fungi have these long hyphal structures, and so that is what allows it to extend. I can actually pull up the picture, it might be helpful here. I believe this was lecture 2, maybe? Or was it 1? Okay, so yeah. So mycorrhiza, remember, is this association between the plants and the fungi because myco means fungi and rhiza means roots. But these fungi have these like structures. So here we see a mushroom and they can form these like finger-like structures that are called hyphae or mycelia, and those connect with the plant's roots. And so that's actually the mechanism by which they can extend the soil and nutrients to the plant through connection to the roots. So that's why it's a mycorrhizal connection or symbiotic association. Yeah.
They don't necessarily like need a fruiting body in order to have like that connection, right? Like they can it just be the mycelia, or does that have to necessarily come from like a mushroom or like?
It usually comes from an actual fruiting body, yeah. So usually it's a fungus, and the fungi have the ability to have these type of things, yeah. It doesn't always have to look like a perfect mushroom like this. It could be, different type of fungi, but the ones that are able to form these hyphae are what causes this symbiotic relationship. All right, and so what else? So the fungi extends the access to the soil. What else does it extend access for the plant? Water, maybe? All right. Cool. So yeah, through the hyphal transport of water. And then what does the plant provide for the fungi? So in all these associations, it's not going to be one benefiting and not the other. There's got to be a beneficial relationship on both ends. So yeah, how does the fungi benefit?
The plant provides energy to the fungus in the form of carbohydrates because the fungus can't carbon fix.
Yeah, exactly. So because the plant can undergo photosynthesis and it can, provide energy through carbohydrates to the fungi. Awesome. So explain the distinction between detection and resolution. Yeah.
Detection is the ability to determine the presence of an object, and resolution is the smallest distance that the objects can be separated by telescopes.
Yep, exactly. So if I show you a broth culture, just like the tubes that we used in lab, if you guys have like a broth culture of media and I tell you there's E. coli in here and you see that it is cloudy in there, which means that there is bacterial growth, you know that there's E. coli in there. Would that be detection or resolution? Detection, yeah, because you can detect with your visible eye that there's something growing in there, same with colonies on a plate, but being able to resolve individual cells requires microscopy. Okay, awesome. So summarize the use of microscopy across many scales of microbial size. So this one is, I think, a little bit more of a big question, but what I'm asking here is there's different scales of microbial size. What types of microscopy will be used for certain sizes? So there's a limit of resolution for different types of microscopes. Like I said, our eyes have a limit of resolution of 0.1 millimeters. So then we need microscopes for anything smaller. So what types of microscopes do we use for the next smallest thing and then the next smallest thing? So that's kind of what I'm asking here. Anybody want to give this one a go?
So I like the largest, I had light microscopy and then scanning electron microscopy, then transmission, electron, and then atomic force, and then X-ray.
Yep, so you've definitely referring to the correct image. So what I'm referring to is this slide right here. And so, yeah, the human eye can resolve anything up to 0.1 millimeters. And then light microscopy, the limit of resolution of using a light microscope, which are just the ones we're using in lab, is 200 nanometers. So these two numbers are important to remember. Anything below 200 nanometers, so smaller and up to just 5 nanometers, will require electrons. So you don't have to remember if it's scanning or transmission, but just electrons bombarding your sample is what will be required for things that are smaller than 200 nanometers up to 5 nanometers. And then anything even smaller than that, you can, like at the angstrom level, you can use X-ray crystallography. I wouldn't worry about this too much, but I think it's important to remember the human eye can detect things at this limit of resolution, and then light microscopy detects anything smaller than what we can see with the human eye, so we need light, and so that the limit of resolution there is 200 nanometers, and then if we have anything smaller than that, then we turn to electron microscopy. Does that make sense? Okay, cool. So describe the 4 different ways that light interacts with an object. Who wants to give this one a try? Yeah.
Absorption, reflection, refraction, and scattering.
Yep. You guys want to tell me what the difference is for each one, define them? Yeah, Kata?
Absorption refers to the phenomena that the object far from the lights. Wait. Sorry, I cannot read myself.
Oh, you're okay. Fine.
Light is part of the light, and then reflection is like what wave from of the light bounces off the surface. Reflection is a light bends when it enters a substance that changes its.
Right, that's refraction.
Small fraction of light.
Yep. So just to summarize, in case not everybody heard that over in the back, absorption is when light is absorbed. So you have a glass slide and you have your light hitting that slide, and so there's going to be light that's hitting it, and so then heat can be generated as one of the things that happens when the light is absorbed. So light's absorbed, heat increases, and because the object or the glass slide in our case is blocking part of that light hitting it. Reflection, just think about, a reflection where you're looking in a mirror. So the wave front of the light bounces off of the surface of the object. So you have this reflection because it's bouncing off. Refraction requires the bending of light. And so remember, that's due to different refraction indices. So light will bend and you'll lose some of that light if the refractive index of your glass slide isn't the same as your media, so that's why we add that oil drop. Refraction also allows us to magnify an image through a lens, so that's also important to remember. Magnification and refraction are related in that way. And then scattering means that a small fraction of the light is just scattered in all directions. So the object size is usually close to the wavelength of the light. So dark field microscopy is one of the ones that utilizes this. All right, so any questions on those 4? All right, number 10, explain the function of bright field microscopy and phase contrast microscopy and the factors that influence them.
I said that bright field generates a dark aura around the object. You have to use immersion oil and use a wider lens. And then phase contrast, there's a difference between the cytoplasm and the medium, and it creates a false 3D.
Yeah, so and then also it's important to note that there's no oil used in phase contrast, and you're not going to the widest lens, so you're stopping at the 40x magnification. But yeah, exactly, everything else that you said was right. All right. Explain the relative advantages and limitations of a wet mount and stain preparation for observing organisms or microbes.
Wet mounts are fast and allow you to observe the cells in their natural habitat without killing them, so you can see them moving. So there's a little contrast with that, and it dries out quickly, so it's hard to.
Yep, exactly. Yep, and then sometimes also microbes can be really quick swimmers, and it's hard to even catch them and see them on the slide, especially if it's a newer culture. In the pond water experiment that I showed you, or that we did last week, unfortunately the microbes, it was an older sample from the river, so they weren't really moving as much, but that's okay. We still got to see them. Nice. And then a stain preparation. Advantages and disadvantages. How about somebody new? Well, it's telling me no signal here. We can still move on while I figure this out. Does anyone want to tell me the advantages and limitations of a stain prep? Yeah.
Staining is where the cells are given a distinct color. And then I mainly talked about the advantages that the stains have markers for their indicating, such as conjugate double bonds and aromatic rings for color, or positive charges that associate with the negative charge of the cell envelope.
Yeah, nice, exactly. So with staining, some of the advantages are you're enhancing the resolution and the contrast because you're fixing the cells and you're staining them so you can see internal structures. And then a disadvantage of staining is that it kills the cells. So that's just one of those things that we do. All right, let me see why this is not working. Turn it off and on. All right. Describe the use of fixation in staining and bright field microscopy. So what's the difference there? How do we fix cells? How do we stain cells?
Fixation sticks them to the slide using methanol or heat, and staining makes them more visible, which we use the saliva.
Yep. Okay, cool. All right. So now we're going into lecture 3. What functions do membrane proteins serve? Yes. Henry.
They serve by adding structural support. They're able to detect for environmental cells, secrete
Virulence?
virulence factors, and ion transport.
Yep. Nice, yeah. So membrane proteins have a lot of functions. They support the structure of the cell, but they can also communicate with the outside of the cell, whether that's environmental signals, if there's something coming in that's trying to harm the cell or that's a toxic substance, then it can secrete virulence factors, which are factors that certain, things can secrete in order to kill off or hinder off the growth of something that's going to be harmful or pathogenic. And so also they're there to help transport ions and energy across the membrane and store energy. All right. I'm really confused as to why this isn't working. All right. Explain how fluidity relates to kinks and cyclic structures within the fatty acids of phospholipids. Yeah.
The kinks increase the fluidity and the cyclic structures decrease them.
Yeah, exactly. So, yeah, I also have written down here that the fatty acids of the phospholipids can change their fluidity to enhance the function of membrane through those things, through the formation of kinks or cyclic structures. And so that's why they're important, because they actually take advantage of this and can change their fluidity by doing these things. Yeah, kinks increase the fluidity and cyclic structures decrease the fluidity. All right, any questions so far? All right. So I asked you guys to draw the structures of a gram-positive and gram-negative cell envelope and compare and contrast them. So does someone want to come up and draw it on the board? Yeah? All right. Well, if like two people want to tackle it, one person can do the gram-positive, one person can do the gram-negative. So how do you guys want to talk through it? What makes a gram-positive cell different than a gram-negative cell? Yeah. You drew it. You should.
I talked about how like the peptidoglycan is on the outside of the cell membrane with the gram-positive.
Yep. Yep. Nice. So, all right. Yeah. So let's see. For the gram-positive, we have this really thick peptidoglycan layer. And then we have the cell membrane here, so that's right. And then what makes this structure important, like what is also in there when we do like the gram stain, for instance, we use the iodine because it creates a complex with a certain feature within this membrane. So anybody remember what that was? Starts with a T. Teichoic acid, yeah. So those teichoic acids are in the gram-positive cell membrane, and they help to really lock in that dye and make it just stick in there. So that's an important one to know, that in the gram-positive membrane, you have these teichoic acids, you have just one cell membrane, and then the peptidoglycan layer, and then do we have an S layer over here? Gives it additional protection. Yeah, so we also have an S layer over here. And then on top of that S layer, we have some glycosyl chains. So these are just some additional features that help, protect and attach to other things. So these are just gram-negative cells. So that's right, yeah, you have an inner and an outer membrane, you have the peptidoglycan layer in the middle. And then, so between the peptidoglycan layer and this outer membrane, there's something that connects this to the cell membrane. Do you know what these are? What that? Yeah, exactly. So lipoproteins. So these we don't have in the gram-positive. So these are things to make sure you know as like differences to compare and contrast. Okay, and then, yeah, and then as well over here, we have the outer membrane. And then do we have an S layer for gram-negative cells? Yeah, so we have an S layer over here. But it's not like the gram-positive where you have the peptidoglycan layer and then the S layer attached to it. There's something in this one that connects the S layer to the outer membrane. So what are these things that connect it? So that would be LPS. It's just another one of those phospholipid chains that just really help to connect the gram-negative cell to other structures, because it's going to have these extra things because it's not as thick as the peptidoglycan layer. So this is LPS, which is similar to those glycosyl chains, but in the gram-positive layer on the outside of the S layer. All right. So, any questions on that? Oh, it worked. Let me just text Kristen. Okay, so why is peptidoglycan a good target for antibiotic therapies? Yeah, exactly. Peptidoglycan is unique to bacteria and not us. And so there are enzymes of making, when bacteria make peptidoglycan, so peptidoglycan synthesis, that can be really good targets for antibiotics. And so peptidoglycan is part of the cell wall, obviously, and so because we don't have a cell wall, it's not going to affect us. If we take antibiotics, we're targeting just the bacteria. What are two mechanisms of resistance that bacteria utilize to evade death or lysis, which just means the bursting of the cell? And so what are the two mechanisms of resistance that bacteria use to evade death by beta-lactam antibiotics?
Mutations and then genetic exchange.
Yep. So there can be gene mutations in the transpeptidase enzyme that will change its shape. And so it can still cross-link and create the cell wall, but now beta-lactam can't fit. So it's important to understand how it works. And then the second one is bacterial gene transfer or genetic exchange. Like I said, bacteria can encounter another bacteria in the environment and they exchange genes. And so they can exchange genes of something called beta-lactamase, which breaks down beta-lactam, so it can target the beta-lactam ring of antibiotics that way. All right, and then lastly, what is the purpose of the S layer in prokaryotes?
I put additional protection in the cell structure.
Yep, exactly. So it's an additional protective layer. It's a crystalline layer of really thick protein subunits. So if you zoom in, I think, let me pull up a picture. Yeah, so over here, it's like, you really see this like crystalline structure that gives it that, protective chain barrier, I guess you could call it, like a fence. But then in addition to that, so it helps contribute to the cell shape but it also helps to protect the cell from desiccation, which is drying out. from osmotic stress. So it helps it from the cell from drying out as well. All right, so any questions on those? All right, everybody feel good about that? Okay. So, now we can go into the remainder of the lecture 4. So it's a shorter lecture. So I wanted to quickly note that the syllabus that I have and like the schedule for the class, it says that we were really going to go through a chapter each time, but I knew that we were going to actually go through a chapter each time, so it's going to be modified, like the content that's actually going to be on the exams. So the first exam is going to be on Wednesday, February 7th. So I think that is a week from tomorrow, or 2 days from now. So, yeah, so for lecture 4, we're basically going to finish chapter 3. So we started off with chapter 3, and so today we're going to finish off with some of the important things in chapter 3. And then we'll have another lecture on Wednesday, and then Monday next week will be a review day, so we can review everything, and then you'll have your exam on that Wednesday. So we're going to continue talking about microbial cell structure and function. All right, perfect. So the 2 sections that I'm going to be reviewing are 3.4 and 3.6, bacterial cell division and specialized structures. So a quick overview, we're going to talk about the nucleoid of bacteria, its structure and how it's expressed, how bacterial cells divide, how specialized structures, including pili and stalks, and all those contribute to cells, and then bacterial flagella and motility. I'm going to try to just see if I can. All right, we could just do it this way. Okay, so, all right, so we talked a little bit about the structure and function of the bacterial cell, starting from the cell envelope. And so here we have a eukaryotic cell, and so we're going to be, and then over here we see the bacteria. So we talked about the cell envelope, but now we're going to kind of work our way more into the middle of the cell and talk more about the nucleus, or the nucleoid, and how it's structured and replicated, and how all of that works. So over here I'm showing you a eukaryotic cell with, in purple, this is the nucleus. It's the nuclear membrane encloses the nucleus, and so it's in purple. And then you have this cell membrane and all of these, the endoplasmic reticulum and all of these different things that are on the outside. But the nucleus is in a compartment on its own. So this is a human cell. And so here we have this E. coli, these cells of E. coli in yellow that are trying to infect this human cell. And so you see that the envelope and the cytoplasm and the nucleoid are all within it over here. So for a long time we thought that prokaryotes were just a bag of soup, or it looked like a bag of soup, but it's really not. There is some organization, but it's not compartmentalized in the same way as eukaryotic cells. And so over here you can see that the nucleoid is actually in these white or clearer areas inside the yellow cells. So they're not encompassed by a membrane, and so it's sharing the same space as the cytoplasm. Right. So, although the nucleoid is not confined by a membrane, it's still organized into domains. So over here I'm showing you these domains. This is an electron micrograph. So the nucleoid, so over here in these clear white spaces, it forms these chromosomal loops called domains, which radiate from the center. So you have the center here, and they kind of come out of the center like this, but they are looped around and they're trying to be condensed, and they have this ordered structure into domains. And so they extend through the cytoplasm. So the DNA and the cytoplasm share the same space. So yeah, here I guess in this image, the bacterial chromosome exploded out when you lose that confinement. So if you were to remove this outer layer that's confining whatever's in there, you're gonna get this like explosion out in these like domains. So now we have imaging types that allow us to see things like that. So that's kind of why I'm showing it to you. And so, yeah, we get this intricate structure that forms once the chromosome is released from the cellular confines. And so it helps researchers really understand that DNA in prokaryotes is still organized in domains, and there's these chromosomal loops. Another way to look at the way that scientists have looked at this is through this fluorescence microscopy. So here they have the same cell. In this one, they have labeled just the nucleoid. And then in this one, they've just labeled the ribosomes that are just within the cytoplasm. And then here they're overlaid on top of each other. And you can really see that the nucleoid and the ribosomes that are in the cytoplasm are in the same space. So yeah, so they actually are contained in the same confines of the cell. All right, any questions so far? So, transcription and translation are coupled. So what does that mean? So when we're just going to briefly go through an overview of transcription and translation. I know you guys have probably gone through this a lot in other classes, but it's important to know that there are differences, really fundamental differences between transcription and translation in bacteria versus in eukaryotes. So generally, we have DNA and we have an RNA polymerase that will transcribe the DNA in transcription, and it'll transcribe it into an mRNA molecule. But in prokaryotes, the mRNA is being made, but then it's immediately bound by a ribosome. Ribosomes are what make proteins. So it'll be bound by a ribosome for translation, and it'll become a polypeptide and a protein. So just some other concepts to remind you that the transfer RNA brings amino acids to the ribosome. And so in prokaryotes, transcription and translation are tightly coupled, and I'll illustrate this in the next slide. So here I'm showing you how prokaryotes, with the absence of this nucleus, transcription and translation are coupled. And so here there's also highlighting some of the antibiotics that can target different parts of this transcription and translation. So just ignore these for now. I won't ask you any questions about these. But just so you know, I talked about how last week we used beta-lactam antibiotics to target the cell wall of bacteria. And so there's also antibiotics that target transcription and translation and other things. So this is just an FYI, but the rest of it isn't. So here what I'm showing you is you have this RNA polymerase that's binding to the DNA strand, and it's going to run along this DNA strand, and it's going to start making this mRNA molecule. So in the blue, that's what we're seeing. So as soon as this mRNA transcript is made, and it's still being continued to be made, there's a ribosome around because everything is in the same part of the cell. It's not confined in different compartments. And so the ribosome that's around is going to immediately jump on the mRNA transcript, even while that transcript is still being transcribed. And so this isn't even being made, but the ribosome can jump on and it can start making the polypeptide sequence and the polypeptide chain for the protein. So it's this coordination of the replication of the chromosome, then transcription and translation, that explains why some bacteria can really divide very, very quickly, some in under than 10 minutes. And so the reason is being that this high coordination of these essential processes, so this is an important take-home message. And so bacteria coordinate DNA replication and gene expression, and eukaryotes separate these processes both spatially and temporally, meaning they're separated spatially within different parts of the cell. We have a nucleus that contains the DNA in eukaryotes, so DNA replication will occur there, and then you'll have the others, transcription, translation happening in cytoplasm, and then temporally, meaning that in eukaryotes, they don't happen simultaneously, but in bacteria, they coordinate them and they are coupled both spatially and temporally. Any questions on that? All right, so cell division, also called binary fission, is the way that the cell will divide, and so we're going to go through some of this. So when there is abundant nutrients in the environment, the cell is almost always replicating its genome while also synthesizing RNA and proteins. And so there's got to be this really highly coordinated growth and expansion of all of the cell parts, from cell division to actual replication of the entire cell. So unlike eukaryotes, like I said, prokaryotes synthesize RNA and proteins continually, while the cell's DNA undergoes replication. So bacterial DNA replication is coordinated with cell wall expansion and ultimately the separation of two daughter cells. So I will go through this. So here we're using an example of a bacteria that has a single singular circular chromosome. So some bacteria will have more than one chromosome, but in these examples, we're going to be talking about ones that have just one chromosome. And so in prokaryotes, the circular chromosome begins to replicate at its origin. So that's this origin of replication site over here, shown in green. And so once it starts to replicate, there are two replication forks that are generated over here, which both start to proceed outwards in both directions. So they always go in opposite directions. So at this region, the DNA starts to become unzipped and forms these replication bubbles that we also call replosomes here in yellow. And so you'll have one origin of replication becoming two, and this forms two replication bubbles and replosomes. Each of these replication forks and replosomes start to move in pairs on both sides. So they move down the left and the right side of the chromosome. And so at this point, it's starting to go bidirectionally, and you start to see this expansion of the DNA trying to loop around. And so what's the other point I wanted to make here? So you have these two replication forks, and at each fork, DNA synthesized by the DNA polymerase and accessory proteins, and so that's what this replosome is in yellow. So each one of these replosomes has two DNA polymerases that start to work on it. So it's important to note that there are 4 total DNA polymerases that are moving in pairs, 2 down the left and 2 down the right. So they're going bidirectionally. So one origin of replication causes 2 replosomes that each have 2 DNA polymerases moving them in either direction. And so notice that just 180 degrees from where the origin of replication is, there's this red sequence here, which is the termination sequence. And so over here, this is the terminator sequence where DNA replication will end. And so there's also a spot, that tells the cell where to stop and where it starts. All right, so what else do I want to talk about here? So yeah, we have these replosomes going down all the way, and they're going to reach this terminator. And so once they reach it, something cool happens. And so here you see that they're starting, as it reaches over here and it's about to start separating, you have these new replication forks forming. So DNA starts another round of replication even before this part ends. And so, like I said, this happens in nutrient-rich environments, so the cells will try to divide as rapidly as possible. And over here, there is this FITZ ring, or FTSZ ring. I'm not gonna go into too much detail about this, but it's a ring that forms in the center of where the septum will form. And so this is needed for the septum to form and for the cell to actually divide into two cells. So we have this Z ring forming, and so it's a GTPase, it's a cool protein that initiates the cell division. So here we see 4 origins of replication. So we have these 4 green dots, and we have these replisomes at each one. So why do we see 4 origins of replication when we only have 1 copy of this terminus? So that's because of the fact that you can get multiple rounds of DNA replication before the cell has even finished replicating the first time. So that's why if you see more than 1, you know that it's a prokaryotic cell and it's just going to continuously divide. So in order to complete cell division, there's going to be, when the replication ends at the termination site, it triggers the growth of the dividing partition or the septum, and so this is where the FTSZ ring will form, and so it forms the septum, it grows inward, and it helps to restrict and seal off the 2 daughter cells. So this is an image of a cocci, so it's just a spherical cell, and so you can see these 2 envelopes really nicely being partitioned. Any questions so far? All right. So that's all you really need to know on DNA replication and how the cell divides. I'm not going to go into too much depth. I think that if you have questions, please let me know. I know that the first time going through it might be a little bit difficult, but beyond that, the only other thing I really want to talk about is just some of these specialized structures. So we already talked about some cellular structures that are found among bacterial world, like the nucleoid, the cell wall. No, I'm good, thanks. Yeah, and so there are the cell membrane, and so I want to highlight a few of the specialized structures that are found in specific bacteria. So some bacteria, like I said in the first lecture, some of them are photosynthetic organisms, so they can photosynthesize. Some of those are cyanobacteria, so that's one example. And so they evolved these extensively folded intracellular membranes. And so these are called thylakoids. So these are also found in the plant chloroplasts for photosynthesis. But essentially, yeah, so bacteria will have these thylakoids similar to plants. And so this is a cyanobacteria that I'm showing you here. And so many of these also have carboxysomes. And so I'm not going to go into everything about carboxysomes because that would be a really long and really fun and really cool lecture. But what you need to know is that they are these polyhedral bodies. They literally are shaped like an octagon. And they're packed with an enzyme called rubisco. And that really helps convert CO2 into sugar for these cells to grow. So just remember that some of the specialized structures among photosynthetic bacteria, like cyanobacteria, are these thylakoids and these carboxysomes that allow it to essentially photosynthesize. All right. Another specialized structure is these pili or fimbriae. So pili or fimbriae, they're used interchangeably. They are these straight filaments of the protein pillin, and they're used in attachment of the cells. So sometimes it can be good for bacteria to adhere to surfaces, and they do this by having these pili or fimbriae. And so there's the little lines sticking out. They're able to attach and adhere to a surface so that the cell doesn't move. There are also stalks, and so these are membrane-embedded extensions of the cytoplasm. So that's another form of surface attachment, but the pili are made of the protein peelin, but these stalks are actually not protein-based, and they are just actually extensions of the cytoplasm. So that's something to remember and to know the difference. So they have these tips over here, and they have, so this is the stalk end of the cell. So this is the stalk, this is the cell. And at the end of the stalk, they make this holdfast. So this is essentially like a drop of glue that it's making, and so that really allows it to adhere to a surface. So that's the secretion, the adhesion factor that they secrete. The stalks are membrane-embedded extensions of the cytoplasm, and pili or fimbriae are protein-based. We also have these nanotubes. They are intercellular connections that help pass material from one cell to the next. So that's one of those examples that I gave you with cells being able to transfer DNA. And so this is one way that they can do it. They can do it by having these intercellular connections. So with cell-to-cell connections that pass the material from one cell to the other. They're actually pretty massive, and they can pass proteins, plasmids, DNA, and other things. Another specialized structure is the rotary flagella. So the ones that are motile are able to move. They generally swim, and these swimmers are able to do this by these rotary flagella. So in a nutrient-filled environment, it's good for the bacteria to stay put, but bacteria often live in these rapidly changing environments, like I've said, and so therefore cell survival often necessitates the ability for these cells to move and relocate. So bacteria are motile through these flagella. And so these flagella are these helical propellers that essentially drive the cell forward. And so there are a few different kinds. And so you don't really have to remember the names, but just to give you an idea, there's some that are monotrichous, which just means that there's one. Mono means one. Amphitrichous means that there's two and that they are on opposite ends of the cell. There is Iophotrichous, which just means that there's a bunch of these flagella and they're all extending from the back. And then these paratrichous cells have the flagella randomly distributed around the cell. So they rotate together in a bundle behind the swimming cell. And so just to give you an idea, so this one is a paratrichous because it has the flagella coming from all around. And so here in this dark field image, you can see that this flagella, so this is what it looks like, but in this cell, it's moving, and so they're kind of all bundled up behind the cell so that it's able to move in one direction. And so if they're all bundled up and they're moving in one direction, let's say counterclockwise, it's going to allow the cell to move in a directed fashion and allow it to swim. But then they can also change the rotation of these flagella so they can now go clockwise, and then that causes them to flail out and tumble, and so they can change the rotation. They can either move towards an attractant or away from a repellent, so they can change the direction of how those flagella rotate. All right. And then the last specialized structure that I want to talk about is this magnetosome. So this is really interesting. It's a membrane-bound organelle that allows bacteria to navigate the Earth's magnetic field. So these magnetosomes, they are magnetotactic bacteria that can align and navigate along magnetic fields. They're found in a lot of aquatic environments. So the example I want to give is the wet mount that we did. A lot of times, if you basically take some of that pond water and put it on a slide and then a cover slip, and then you take a magnet and you like put the magnet on there, you can like see the cells moving towards or away the magnet, because they literally have like this ability to I guess, yeah, just align and navigate along the geomagnetic field of the Earth. And so This is important for them because it allows the cells, especially in aquatic ecosystems where there's very low oxygen, so they're either anaerobic or microaerophilic environments, which just means that they need low oxygen environments. These cells find these low oxygen environments by following the magnetic field of the Earth, and so they can go there that way. And so, yeah, it's really magnetite that's biomineralized in these membrane-bound organelles. And so how does this happen? Essentially what they do is they form this compass needle inside of the cell. So they're organized by a filament to form an intracellular compass needle. And so I just think that that's really cool. That's one of those things that we recently discovered about bacteria. So any questions about this?
What kind of stains are these?
Sorry?
What kind of stains are these?
So this is a regular phase contrast microscopy. This one is more of like a 3D structure. This isn't a stain. I think this is just something that a computer like generated, which is really cool. And this one, I believe it is stained. They probably just use fluorescence microscopy and stain the cytoplasm and this green fluorescent protein and the magnetosomes. They probably, I don't know, stained it with some like red or something, but yeah, that's probably what they did. I'm not exactly sure, but these images are from the book. I'm sure that they probably will have it somewhere in the textbook to show you what exactly the type of microscopy and stain they used. So yeah, so you don't have to remember all of these, but yes, bacteria do have organelles because I want to emphasize this because it's wrong in previous textbooks. And whether you have or don't have organelles is no longer a difference between prokaryotes and eukaryotes, like we all have learned. And we now know that bacteria have a diversity of organelles. They come in a variety of different flavors. The four that you need to know, you don't have to know the actual subheadings here of what they are, but they can be membrane bound. And so, you know, we talked about magnetosomes, so that's what it falls under, the membrane bound class of organelles. You don't have to remember that though. Some are protein bound, some are lipid monolayers, some are phase defined. But what's important to remember is that all of these organelles increase the metabolic efficiency of the cell and or allow it to sequester specific, often toxic compounds. So if something's toxic to the cell, it helps to sequester it. And so here's just some pictures that you can look at. Don't have to remember any of these, but just gives you examples of what each of these could look like. And to end here, we'll do one concept check. So what is the extension of the cytoplasm that attaches bacteria to a surface cult? So remember, we're talking about an extension of the cytoplasm. Yeah.
A stalk?
Yep, exactly. So a stalk, because remember the main difference is that the pili or the fimbria are all protein-based, and so a stalk is the membrane enclosed, is the extension of the cytoplasm. So A, B, and C are all membrane-based. And nanotubes don't adhere to a surface. What they do is they physically connect cells to one another. So with that should be all, and I will see you guys on Wednesday. Let me know if you have any other questions.